So, let's go. Number 1. Deinocroton draculi, the terrible tick. Picture a single drop of tree resin falling roughly 99 million years ago in a humid forest in what is now northern Myanmar. Trapped inside it is a tick, its body swollen to nearly 8 times its normal size, bloated with a meal of blood, and clutched right beside it, a feather. Not the feather of any bird you would recognize, but the feather of a dinosaur. This is the closest the fossil record has ever come to the opening scene of a monster movie, and it is completely real. The specimen was described in 2017 in the journal Nature Communications by Enrique Peñalver, Ricardo Perez de la Fuente, and David Grimaldi, working with Burmese amber from Kachin State. The amber dates to the mid-Cretaceous, a period called the Cenomanian. The name they chose says everything. Deinocroton means terrible tick. The species name, Draculi, is a nod to Dracula himself. A related amber tick, Cornapalpatum burmanicum, was found physically tangled in a dinosaur feather, while 2 Deinocroton specimens were preserved together, one of them grossly engorged. The feather belonged to a dinosaur on the branch leading toward birds, not a modern bird, because modern birds had barely begun to diversify. Now, you might be hoping this is where we extract ancient DNA and grow a dinosaur. It is not. Amber does not preserve usable genetic material over these spans, and the blood inside the tick is mineralized, not liquid. But the ticks carried another clue. Tiny hair-like filaments stuck to their bodies turned out to be the bristles of skin beetle larvae, insects that today live in the nests of animals. That suggests these ticks shared a nest with their feathered hosts, creeping out to feed and crawling back, exactly as nest-dwelling ticks do now. Ticks are not insects. They are arachnids, with 8 legs as adults, and they are obligate blood feeders for their entire lives. Modern ticks carry Lyme disease, babesiosis, and anaplasmosis, and while we cannot know what this one carried, the machinery for blood-borne infection was clearly in place. There is an ethical shadow over Burmese amber, which comes from a conflict zone, and many journals now restrict new specimens. Deinocroton itself is extinct, a dead-end branch with no known descendant. So here is the question that drives this entire video. If a tick was already riding dinosaurs, how far back does parasitism really go? Some researchers think ticks arose deep in the Paleozoic. Others argue for a Mesozoic origin alongside their hosts. To answer it, we have to go much further back than the dinosaurs. Number 2. Invavita Piratica, the parasite caught in the act. Travel back to 425 million years ago, to a Silurian seafloor in what is now England. Here, in a deposit called the Herefordshire Lagerstätte, paleontologists found something almost no fossil ever shows us, a parasite still gripping its host at the moment of death. The find was described in 2015 in Current Biology by David Siviter, Derek Briggs, Derek Siviter, and Mark Sutton. They named it Invavita Piratica. Invavita means attached to life. Piratica means piracy. The host was an ostracod, a tiny crustacean that lives inside a hinged shell like a seed of the species Nymphatalina gravida. Clinging to it were as many as a dozen small, soft-bodied parasites. And that softness is exactly why this fossil should not exist. Soft tissue almost never survives. The Herefordshire deposit is special because volcanic ash settled over marine animals and preserved them as voids in calcite inside hard nodules. To study them, the team used serial grinding tomography. They ground the fossil away in microscopically thin slices, photographing each layer, then rebuilt the whole thing as a digital 3-dimensional model. The method destroys the original fossil completely, but it is the only way to see inside the rock. What they saw was remarkable. The host ostracod was preserved, carrying eggs and possibly brooded young, so the researchers could even ask whether the parasites were harming its ability to reproduce. The parasites sat near the host's feeding currents, in a position to steal food particles, a strategy called kleptoparasitism, or possibly to feed on the host's own tissues. Parasitism is one of the most common ways of life on Earth today, and yet it almost never fossilizes, because it leaves so little hard evidence behind. This single slab is one of the oldest direct records we have of one animal living off another, frozen mid-act. Ostracods are still everywhere in the oceans, and they still carry parasites, so the relationship has deep roots. The Silurian itself was a world recovering from the end Ordovician extinction, a time of spreading reefs, the rise of jawed fishes, and the first plants creeping onto land. The species is extinct and has no named living descendant. There is still debate about what exactly it was. Some see a true parasite draining its host. Others suspect a commensal that merely stole scraps. A few wonder if it was simply a hitchhiker. Whatever the answer, it tells us parasitism was already an ancient art 425 million years ago, and the next freeloader stuck around even longer. Number 3. Platyceras, the snail that lived on waste. Some parasites are terrifying. This one is just deeply, memorably gross. Platyceras was a snail that lived across the Paleozoic, from the Silurian through the Permian, a span of well over 100 million years. And for that entire time, it did one thing extraordinarily well. It sat on the rear end of a sea lily. Sea lilies, properly called crinoids, are not plants. They are animals, relatives of starfish and sea urchins, anchored by long stalks with feathery arms that filter food from the water. Their digestive waste exits near the top of the body, through a vent on the upper surface called the tegmen, and that is precisely where Platyceras parked itself, cementing its shell directly over the crinoid's anal opening to consume the steady supply of waste, a habit called coprophagy. Researchers argue about what to call this relationship. Some say parasitism, some say kleptoparasitism, the theft of another animal's food, and some say it was merely commensal, harmless freeloading. But there is a strong clue it crossed the line into parasitism. Many crinoid fossils showed deformed or swollen growth directly beneath the attached snail, the host's body visibly reacting to a long-term tenant. The fit between them was remarkably tight. The shape of the snail's shell opening often matches the contours of the crinoid's surface exactly, evidence of a relationship refined over generations. Some researchers go further and suggest the snail may have rasped directly at the crinoid's tissues, not just waited for waste. In the Paleozoic, shallow seafloors were covered in vast sea lily meadows, providing a near-endless supply of hosts. The pairing is so consistent that paleontologists treat an attached Platyceras as a diagnostic feature when they study certain fossil beds, especially the Carboniferous limestones of North America and Europe. The snail had a low, cap-like shell, perfectly suited to clamping down on a flat surface and staying put. Both the snail and the crinoid were heavily mineralized, which is why this is one of the rare parasite relationships preserved beautifully in hard parts, rather than guessed at from faint traces. Platyceras is extinct, and crinoids today survive in far smaller numbers than at their Paleozoic peak, though modern crinoids still host snails and worms in an echo of the ancient arrangement. The fact that this partnership lasted across multiple geological periods suggests it was stable, not quickly lethal. So was Platyceras a thief, a parasite, or a tolerated guest? The honest answer is that it may have been all three at different moments. And from a snail on a seafloor, we now climb up onto the dinosaurs themselves. Number 4. The giant Jurassic dinosaur fleas. If the idea of a flea does not frighten you, it is because you have never seen one the size of your thumbnail, with a mouth like a saw. In the Middle Jurassic and early Cretaceous of China and Russia, there lived a group of large, wingless, blood-feeding insects that paleontologists nicknamed the dinosaur fleas. The best known belong to the genus Pseudopulex, including Pseudopulex jurassicus and Pseudopulex magnus, described around 2012, by Diying Huang and George Poinar, among others. A female Pseudopulex magnus reached about 20.6 millimeters, which is roughly 2 centimeters, around 10 times the length of a modern flea. These were monsters by parasite standards. Their bodies were flattened, ideal for sliding between feathers or scales, and their legs ended in long, curved claws built to grip onto skin and cling tight. Most striking of all were the mouth parts, a long, stout, saw-edged proboscis designed to pierce through thick hide and draw blood. A related genus, Sauropterus, comes from the early Cretaceous of Russia and was originally described by Alexander Rasnitsyni in 1992, possibly as a parasite of pterosaurs, the flying reptiles. The name translates roughly to lizard louse, reflecting that early interpretation. Crucially, these insects lacked the powerful jumping legs of true modern fleas, so they could not leap. They crawled across their hosts instead. Their exact place in the family tree is debated. Some researchers group them in a stem flea lineage called Pseudopulicidae, while others treat them as a separate extinct experiment in blood feeding. What is clear is that their large size suited large hosts with tough skin, exactly the kind of animal walking around in the Jurassic. The fossils come from the Daohugu and Jehol Lake deposits of China, the same rocks that preserve feathered dinosaurs and some of the earliest mammals, so plausible hosts were close at hand. These are compression fossils, flattened in fine lake sediment, yet they preserve the body outline and the menacing mouth parts in fine detail. Modern fleas, as we know them, would not appear in recognizable form until much later, which means these creatures represent an entirely earlier attempt at the same parasitic lifestyle, tens of millions of years ahead of schedule. There is honest disagreement about whether every one of these giant fleas fed on dinosaurs specifically, or whether some targeted mammals or pterosaurs instead. They are all extinct, with no direct modern descendants. But the image is unforgettable. A 2-centimeter insect with a saw for a face, crawling across the back of a dinosaur. And the next parasite was caught with its last meal still inside it. Number 5. Megamenopon rasnitsyni, the louse full of feathers. Imagine a parasite preserved so perfectly that you can see what it ate for its final meal, still packed inside its gut. That is Megamenopon rasnitsyni, a fossil louse described in 2004 by Torsten Wappler, Vincent Smith, and Alexander Rasnitsyni from the Eckfeld Mar in Germany. It dates to the Middle Eocene, around 44 million years ago. This is a chewing louse, a bird louse, a member of the order Pteroptera, the same group that infests birds today. And inside its preserved gut are the barbules of feathers, the microscopic branches that zip a feather together. That is direct physical proof of what this insect ate. It was a feather eater, an ectoparasite living in the plumage of a bird. The louse is fairly large, several millimeters long, comparable to the lice that live on big modern water birds, and its anatomy closely resembles a group called the Amblycera that infests ducks and similar birds. So the host was very likely something duck-like or rail-like. The deeper significance is that feather lice were already highly host-specialized by the Eocene, not generalists feeding on whatever they found. Lice are flightless and spend their entire life cycle on the body of their host, which makes fossil lice extraordinarily rare. You essentially need the host and the parasite to die and be preserved together under perfect conditions. The Eckfeld and Messelmar lakes provided exactly that, volcanic crater lakes with oxygen-starved bottoms that preserved insects and even bird feathers in astonishing detail. Bird fossils with intact plumage from the same deposits confirm that suitable hosts were present in the environment. Louse evolution is tightly linked to host evolution, which makes fossils like this priceless for calibrating molecular clocks, the genetic estimates of when lineages split. Modern lice come in two broad types: chewing lice that eat feathers and skin, and sucking lice that drink blood. Megamenopon is firmly a chewer. The preservation of feather barbs in its gut makes it one of the most informative fossil parasites ever found. The species is extinct, an early branch of the feather louse family tree. The theme it illustrates, the way parasites and hosts evolve in lockstep, called co-speciation, runs through all of modern parasitology. The Eocene was a warm greenhouse world of lush forests and abundant bird life, and lice that cemented their eggs to feathers, much as they do today, thrived in it. So far, our parasites have clung to the outside of their hosts. The next one lived deep within a gut, and it is far older than any tapeworm we knew about before. Number 6. The 270-million-year-old shark tapeworm. Tapeworms are almost impossible to fossilize. They are soft. They live hidden inside a gut, and when their host dies, they decay with everything else. So for a long time, the fossil record of tapeworms was essentially blank. Then, in 2013, a study published in the journal PLOS One changed that by finding tapeworm eggs inside fossilized feces. The work was led by Paula Dencian-Diaz, with George Poinar among the authors, and the specimen came from the Rio do Rasto Formation in southern Brazil. The fossil dung, called a coprolite, dates to the middle to late Permian, roughly 270 to 260 million years ago, long before any dinosaur existed. The coprolite was spiral-shaped, the telltale sign of a fish with a spiral valve in its intestine, a feature found in sharks and their relatives. Inside it, the researchers found a cluster of around 93 eggs, attributed to a tapeworm, a cestode, that had lived in the gut of a Paleozoic shark. Several of the eggs preserved internal structure, and at least one appeared to contain a developing larva, an almost unbelievable level of detail for something so small and so old. The eggs measured only tens of micrometers across and had to be studied in thin sections under a microscope. This single find pushed direct evidence of tapeworm parasitism back by roughly 140 million years compared to anything known before. It is a perfect demonstration of why coprolites are so valuable to the field of paleoparasitology. The worms themselves never fossilize, but the eggs and cysts they release get trapped in dung, sediment, and the right chemical conditions, leaving a record where the animal itself leaves none. The Permian was the era just before the largest mass extinction in the history of life, the end Permian event, sometimes called the Great Dying. Sharks and their kin were diverse and abundant in both the seas and the freshwater systems of the time, giving tapeworms plenty of hosts. Modern tapeworms infect virtually every group of vertebrates, including us, usually through complex life cycles that pass through intermediate hosts. This fossil shows that such complex cycles, using fish as hosts, were already in place hundreds of millions of years ago. The eggs even resemble those of modern tapeworms, a sign of just how deeply conserved their reproductive structures are. It is worth stressing that this is entirely distinct from the human tapeworms we picked up much more recently from carnivores and livestock. This ancient cestode species is extinct, even as tapeworms as a group continue to thrive. And the next parasite is so strange that scientists could not even agree on what kind of animal it was. Number 7. The Cretaceous tongue worms in amber. Some parasites are disturbing because of where they live. Tongue worms, properly called pentastamids, live inside the respiratory tracts of vertebrates, especially reptiles, lodged in the lungs, the airways, and the nasal passages. And for a long time, nobody was sure what they even were. They look like segmented worms, but they are not worms. They are not insects either. Genetic studies and the structure of their sperm now place them as highly modified crustaceans, distant relatives of crabs and shrimp that abandon the open ocean to live inside other animals' breathing systems. Starting around 2017, researchers reported pentastamids preserved in Cretaceous Burmese amber, about 99 million years old, and at least one specimen was found near or attached to a lizard, an actual host caught alongside its parasite. This is extraordinary because pentastamids live deep inside their hosts, which means they almost never end up preserved externally, where amber can catch them. The amber captured the parasite's annulated, ringed body and the hooks around its mouth that it uses to anchor inside an airway. The name pentastamid means 5 mouths, from the appearance of the central mouth flanked by 2 pairs of hook-bearing structures, which early naturalists mistook for extra mouths. Today, these parasites infect snakes, crocodiles, and lizards, and occasionally they infect humans, causing a disease called porocephalosis or pentastomiasis. Their fossil record is exceptionally sparse. Most known fossils are isolated, tiny larvae from Paleozoic marine rocks, including specimens from the Cambrian and Ordovician of Sweden, which suggests the group originated in the sea long before it moved into land-living vertebrates. The Cretaceous amber finds are so valuable because they help bridge an enormous gap between those ancient marine larvae and the modern parasites of reptiles. The lizard host trapped in the same amber tells us pentastamids were already targeting reptile respiratory systems by the Cretaceous. Burmese amber is famous for preserving small vertebrates, including lizards and even a hatchling bird, which gives researchers rich context for the kinds of animals these parasites lived alongside. Pentastamids have a complex life cycle that sometimes passes through an intermediate host before reaching the final reptile, much like other parasites we have met. The discovery that this strange creature is actually a crustacean was one of the genuine surprises of modern zoology. These amber species are extinct, though the lineage continues inside reptiles today, and the same ethical caveats about Burmese amber apply. The fact that even an internal parasite occasionally makes it into the fossil record shows just how much the right conditions can preserve. And our next parasite was riding inside an insect, carrying a disease that still kills hundreds of thousands of people every year. Number 8. Paleohaemoproteus burmacis, the malaria of the dinosaurs. Malaria feels like a modern enemy, but its family tree is staggeringly old. In 2005, George Poinar and colleagues described Paleohaemoproteus burmacis, a malaria-type parasite preserved inside a biting insect trapped in Burmese amber roughly 100 million years ago. The parasite is a hemosporidian, the same broad group that includes Plasmodium, the genus that causes human malaria, and Haemoproteus, which infects birds and reptiles. It is interpreted as an ancestor of those blood parasites. What makes the find especially interesting is the carrier. The vector was not a mosquito. It was a ceratopogonid biting midge, a tiny blood-feeding fly, which tells us that early malaria-type parasites were using midges before they ever colonized mosquitoes. Poinar argued that these parasites most likely infected reptiles of the time, and possibly even dinosaurs, and he laid out the broader idea in a popular book written with Roberta Poinar called What Bugged the Dinosaurs, suggesting that insect-borne diseases added to the ecological stresses dinosaurs faced. Inside the amber insect, the researchers identified preserved stages of the parasite resembling oocysts and sporozoites, the developmental forms that malaria parasites pass through inside their insect vectors. A separate younger find named Vetufibrus ovatus was reported in 2011 from a fossil fly in Dominican amber of Miocene age, showing the lineage carried on through later epochs. The stakes are not abstract. Today, human malaria is transmitted by Anopheles mosquitoes and is responsible for hundreds of thousands of deaths every year. Bird malaria, caused by Plasmodium relictum, devastated native Hawaiian birds after mosquitoes were introduced to the islands, driving some species toward extinction. This Cretaceous fossil suggests the malaria lineage is at least 100 million years old, far older than humanity, and molecular clock estimates broadly agree on a Mesozoic origin. It is important to be precise, though. The claim that these parasites infected dinosaurs is an inference, because the parasite was found inside the insect, not inside a dinosaur. Biting midges are abundant in Cretaceous amber, consistent with a rich community of blood-feeding insects, But the idea that disease helped doom the dinosaurs remains controversial and is not accepted as a primary cause of their extinction. What the fossil does provide is a firm minimum age for vector-borne blood parasitism, proof that the two-host life cycle, alternating between a biting insect and a vertebrate, was running deep in the age of reptiles. The genus Paleohaemoproteus is extinct, while its modern relatives thrive in birds, reptiles, and mammals. And speaking of insects with a belly full of blood, the next fossil is the one that inspired a movie. Number 9. The blood-engorged fossil mosquito. Everyone knows the premise. A mosquito drinks dinosaur blood, gets trapped in amber, and millions of years later, scientists pull the blood out and clone a dinosaur. It is a brilliant story, and the real fossil that comes closest to it shows exactly why the story can never come true. In 2013, Dale Greenwalt and colleagues at the Smithsonian reported, in the Proceedings of the National Academy of Sciences, a fossil mosquito with its abdomen visibly swollen, full of a preserved blood meal. The fossil is not in amber at all. It is a compression fossil, flattened in fine shale from the Kishinane Formation in northwestern Montana, and it dates to the Middle Eocene, around 46 million years ago. The team analyzed the distended abdomen and detected iron and heme, the molecule that carries oxygen in blood. This was the first fossil mosquito ever found preserving the chemical traces of an actual blood meal inside its body. They used mass spectrometry to identify the porphyrin and heme compounds, confirming that what filled the abdomen really had been blood. But here is the crucial part. There was no DNA, and there could not be. Genetic material breaks down within thousands of years, not tens of millions. So the dream of extracting intact dinosaur DNA from a preserved blood meal is simply impossible. This fossil is frequently cited precisely to debunk that idea. The specimen is a female, which fits, because only female mosquitoes take blood meals, using the protein to develop their eggs. The fossil came to the Smithsonian as part of a large collection gathered by amateur fossil hunters, a reminder of how much science depends on dedicated collectors. The Kishinane lake sediments are fine-grained enough to preserve even delicate insects, but the odds against this particular fossil are staggering. A blood-filled mosquito is fragile and easily crushed. For one to settle gently into sediment without bursting, and for the blood chemistry to survive 46 million years, is astronomically unlikely. Heme turned out to be far more stable over deep time than anyone expected. The host animal whose blood this was remains unknown, since no DNA or species-specific protein survived to identify it. The find confirms that blood-feeding mosquitoes existed and behaved much as they do today by the Eocene, and modern mosquitoes carry malaria, dengue, Zika, yellow fever, and West Nile virus, making them the deadliest animals on Earth to humans. Mosquitoes belong to the family Culicidae and have a fossil record reaching back into the Cretaceous. This particular species is extinct, one of countless lost blood feeders preserved by sheer chance. And for our final parasite, we turn to the most famous dinosaur skeleton in the world. Number 10. The parasite that ate holes in Sue the Tyrannosaurus. In the Field Museum in Chicago stands Sue, the largest and most complete Tyrannosaurus rex ever found. And in Sue's lower jaw are several smooth, round holes. For years, those holes told a dramatic story. They were read as bite wounds, scars from savage battles between tyrannosaurs tearing at each other's faces. Then, in 2009, a study in PLOS 1 by Ewan Wolfe, Stephen Salisbury, John Horner, and David Varricchio offered a very different explanation, one that is far more unsettling. They argued that the lesions closely resemble the damage caused by trichomoniasis, a disease produced by a single-celled parasite called Trichomonas gallinae. This parasite infects modern birds. Pigeons and birds of prey suffer from it, where it is known as frounce or canker, and it erodes the tissues of the throat and the underlying bone, leaving smooth-edged holes. Since birds are living dinosaurs, the direct descendants of the theropod lineage, a shared parasite-driven disease across tens of millions of years, is entirely plausible. The key evidence is the shape of the holes. Their smooth margins do not match the rough, crushed edges that a powerful bite would leave when teeth punch through bone. And Sue is not alone. Several other tyrannosaur specimens show similar lesions, which points to a recurring disease rather than a series of coincidental injuries. In severe cases, trichomoniasis can swell and block the throat entirely. The researchers suggested that badly infected tyrannosaurs may actually have starved to death, unable to swallow, brought down not by a rival, but by a microbe. The parasite could have spread through cannibalism or through the face-biting behavior that tyrannosaurs are known to have engaged in. The parasite itself, of course, is not fossilized. The entire diagnosis rests on the pattern of damage in the bone, read against the diseases of living birds. Trichomonas is a flagellated protozoan that lives in the digestive and respiratory tracts of its hosts, and the hypothesis, while widely discussed, remains debated, with some researchers favoring other infections or simple injuries. Sue was discovered in 1990 in South Dakota by the fossil hunter Sue Hendrickson and was acquired by the Field Museum in 1997 for more than $8 million. This reinterpretation transformed the story written in those holes, from a tale of combat into a tale of disease. It is one of the most accessible examples of paleoparasitology there is, because the host is a famous, beloved, instantly recognizable dinosaur. The ancestral protozoan that may have infected the tyrannosaurs is, as a Cretaceous lineage, extinct, even as its relatives persist in birds. And it leaves us with a humbling thought that even the most fearsome predator that ever walked was, in the end, prey to something it could not see. Step back and look at what these 10 fossils have in common, and a striking pattern emerges. Every single one of them is a miracle of preservation, because parasites are almost designed to vanish from the fossil record. They tend to be small, they are often soft-bodied, and many of them live hidden inside their hosts, where decay erases them along with everything else when the host dies. To find any of them at all, nature has to cheat in one of a handful of specific ways. The first is amber, fossilized tree resin, which entombs small creatures so rapidly that it captures their external anatomy in 3 dimensions, every leg segment and mouth hook intact, as it did with the tick, the tongue worms, and the malaria-carrying midge. But amber preserves no usable DNA. The second is coprolites, fossilized feces, which trap the eggs and cysts that soft worms release, leaving a record of a parasite that itself rots away, exactly as the Permian shark tapeworm was found. The third is the Great Lagerstätten, rare deposits like Herefordshire, Messel, and Kishinev, where fine-grained sediment and oxygen-starved water preserve soft tissues in extraordinary detail, giving us the 425 million-year-old Invavita and the feather-filled Louse. The fourth is chemical fossils, the heme and porphyrins detected by mass spectrometry in the blood-filled mosquito, molecules that outlasted every cell around them. And the fifth is trace fossils, the borings, deformations, and lesions that record a parasite's presence even when the parasite is entirely gone, like the swollen crinoids beneath Platyceras and the holes in Sue's jaw. Each method shows us a completely different slice of parasite life, and each is rare. Sometimes seeing the fossil even means destroying it, as the grinding tomography that revealed Invavita did. What this really tells us is sobering. The fossil parasites we know are a vanishingly small fraction of those that once existed. The record is biased toward parasites that happen to associate with hard parts, or that died in the few places on Earth where soft tissue could survive. For every parasite we have found, untold millions left no trace at all. Which raises a natural question. If parasitism is this old and this pervasive, where exactly did it begin? Lay these fossils out along a timeline, and they sketch the deep history of parasitism itself, a story far older and far more relentless than most people imagine. The oldest of our examples, Invavita Piratica, takes us to the Silurian, 425 million years ago, and shows that parasitism was already established among marine invertebrates, animals living off other animals in the ancient seas. Move forward into the rest of the Paleozoic, and you find Platyceras riding its crinoids for over 100 million years, and the marine larvae of early tongue, worms drifting through the same oceans. By the Permian, 270 million years ago, the shark tapeworm shows that complex internal parasites with multi-stage life cycles were already thriving inside vertebrate guts. All of this before a single dinosaur had evolved. Then comes the Mesozoic, the age of dinosaurs, and parasitism explodes across the new hosts. Giant fleas crawl across dinosaur skin, terrible ticks ride feathered theropods, biting midges carry the ancestors of malaria, and tongueworms colonize the airways of lizards. After the dinosaurs fall, the Eocene gives us the feather-eating louse and the blood-filled mosquito, parasites that look and behave essentially like their modern counterparts on birds and mammals. The lesson in this sequence is profound. Parasitism is not a single invention that happened once. It is a strategy that life reinvents again and again in the sea and on land, on invertebrates and dinosaurs and birds, because living off another organism is simply one of the most reliable ways to make a living. Every host that evolves becomes a new world to be colonized, and an evolutionary arms race begins, host defenses sharpening against parasite tools across millions of years. Mass extinctions reset the cast of hosts, but parasitism always rebounds. Vectors shift, midges giving way to mosquitoes, while the parasite lineages themselves persist. Biologists today estimate that a very large fraction of all species on Earth are parasitic in some way, and that essentially every free-living species hosts parasites of its own. Seen through deep time, parasitism is not an aberration or a horror. It is a fundamental feature of life, woven into the living world from almost the beginning. Which brings us back to the promise in the title. We called these the parasites you are glad are extinct. But is that even true? Here's the uncomfortable truth hiding inside this whole video. Yes, every specific creature we have met is extinct. Deinocroton, the terrible tick, the giant Jurassic fleas, Platyceras, the waste-eating snail, the Permian shark tapeworm, the amber tongueworms, Paleohaemoproteus, the dinosaur Malaria. All of them are gone as species, but their lineages, their families, the strategies they pioneered are very much alive. And in many cases, they are more dangerous to us now than their ancient versions ever were. Ticks did not vanish with Deinocroton. They spread Lyme disease and a growing list of other infections, and they are arguably more medically important to humans today than at any point in the past. Fleas did not disappear when the giant Jurassic forms died out. Their descendants carried Yersinia pestis, the plague, which reshaped human history and killed tens of millions of people. Tapeworms still infect billions of people worldwide through undercooked meat and fish. Malaria, the descendant of that Cretaceous lineage in amber, still kills hundreds of thousands of people every single year. Lice remain ubiquitous on birds and mammals, including us. Even tongueworms still infect people in parts of Africa and Asia, causing porocephalosis. So when we say we are glad these parasites are extinct, we are telling a comforting half-truth. The individual species are gone, but the strategy they represent is thriving, and its modern forms are often worse. In fact, several of the ancient parasites may have been less dangerous to humans than their living relatives, simply because they were not adapted to us at all. The genuinely, comfortingly extinct ones are the oddities, the 2-centimeter fleas, the specific amber species, the snail that sat on sea lilies. Those particular nightmares really are over, But the deeper lesson is an irony worth sitting with. We feel a shiver of relief at these extinct parasites while we share our world, our homes, and sometimes our bodies with their direct descendants. So how does modern science actually read these ancient invaders? And what can the dead teach us about the diseases of the living? The study of extinct parasites is not a museum curiosity. It is an active, surprisingly high-tech science, and it feeds directly into how we understand disease today. One of its most important tools is the molecular clock, the technique of estimating when lineages diverged by counting genetic differences and converting them into time. But a molecular clock is useless without calibration, without fixed points in real time, and that is exactly what fossils like the Eocene louse and the Cretaceous Malaria midge provide. They anchor the genetic estimates to actual dates in the rock record. Alongside that, the field of paleoproteomics has grown powerful, using mass spectrometry to recover ancient molecules like the heme preserved in the Montana mosquito, reading chemistry where no cells survive. Microcomputed tomography, a kind of high-resolution X-ray scanning, now lets researchers peer inside amber and coprolites without destroying them, a gentler alternative to the grinding tomography that revealed Invavita. All of this matters beyond the fossils themselves. Understanding how parasites and their hosts have tracked each other across deep time, the studies of co-speciation help scientists model how diseases emerge, jump between species, and spread. Knowing that the malaria lineage is 100 million years old reframes how we think about its adaptability and our chances of defeating it. The collapse of native Hawaiian bird populations under introduced bird malaria stands as a modern warning of what happens when a parasite meets hosts with no defenses. There is also a growing recognition among ecologists that parasites are not simply villains to be eradicated. They are integral parts of ecosystems, regulating populations and shaping food webs, and the loss of parasite diversity can itself be a sign of an ecosystem in trouble. The field is even reckoning with its own ethics, as the troubling origins of Burmese amber in a conflict zone reshape how and whether new specimens are studied. And with every new amber drop split open and every coprolite sectioned, the dates keep getting pushed back, a reminder of how incomplete the record still is. Much of this progress rides on museum collections and the patient work of amateur collectors, the same kind of hands that delivered the blood-filled mosquito to the Smithsonian. Which leaves one last thing to consider: what all of this really means. Run the whole journey back through your mind for a moment. We began with a bloated tick locked in amber, gripping a dinosaur feather 99 million years ago, and we traveled all the way back to a parasite clinging to its host on a Silurian seafloor 425 million years ago, then forward again through Permian shark guts, Jurassic dinosaur skin, Eocene lakes, and finally, the jaw of the most famous Tyrannosaurus on Earth. What emerges is not really a gallery of monsters. It is a portrait of life, as it has always been. The most magnificent and fearsome creatures that ever existed, the feathered dinosaurs, the great sharks, the towering tyrannosaurs, were never alone. They carried passengers, tenants, and invaders, animals that fed on their blood, lived in their airways, ate their feathers, and sometimes killed them from the inside. Even Sue, the apex predator of her world, was almost certainly brought low by something far too small to see. Parasitism, it turns out, is not a footnote to the history of life. It is one of its central plots, a strategy reinvented in every age, on every kind of host, across hundreds of millions of years, and the fossil record only ever shows us fragments of it. The rare drops of amber, the lucky coprolites, the few places where soft tissue could survive, each one a tiny window into a vast and mostly invisible world. The species we have met tonight are gone, genuine extinctions, and there is a strange comfort in that. But the strategy they embodied lives on in every tick, flea, worm, and microbe that shares the planet with us. Perhaps the right response to all this is not disgust, but a kind of wonder. The recognition that parasites are as much a part of the story of life as the giants they rode upon. The next time someone splits open a drop of amber or sections a piece of fossil dung, there is no telling what ancient passenger might be waiting inside. Patient, preserved, and ready to tell us a little more about the hidden half of life on Earth.